After you learn about the different type of organic molecules in our body, it's time to learn a little bit more about the enzymes and how the enzymes are related to the use of energy in our body. By structure, an enzyme is a protein, is a type of protein with a specific function. What is the function of the enzyme? It's a catalyst. And by definition, a catalyst is a substance that increases the rate of that chemical reaction without itself undergoing any permanent chemical change. We mentioned that the proteins can be built up or can be break down. The enzyme, when it's produced, is ready to perform the function, and the function is a catalyst, means that any chemical reactions that are using an enzyme can proceed in a faster rate or in a faster speed, but also with the use of less amount of energy. That's why I call them that they are energy saver. Certainly increase the rate of reaction and never change the structure. Like the enzyme is using particular molecules that are called substrates, and that substrates or molecules use this enzyme to produce a totally new product. But important to know that they lower the activation energy of the reaction, means that that chemical reaction can proceed with less amount of energy. The enzymes also has different characteristics that are common characteristic for all the type of all the different enzymes we have in our body. They have a conformation. They have a special 3D shape for any single enzyme we have in our body. They are not similar between each other in terms of that particular 3D shape. All of them, they have an active site. The active site is a special place within the enzyme that is used to produce this conversion that is used by the substrate or reactants in order to produce a totally new product. All the enzymes also use substrates, also called reactants. What is a substrate? Could be is a molecule, and this substrate could be only one or could be more than one. It doesn't matter, but it's a product used or a molecule used by the enzymes to be converted in another totally different molecule called a product. All the enzymes also responds to a lock and key model. That specific shape of the substrates fit perfectly like a glove in a hand with that particular enzyme. Lock and key model. The substrate has a specific shape that fit like a glove in that active site. Also, they, in some cases, in several cases, they are induced fit model. Means that when that substrate doesn't fit perfectly in that active site, they have the capability to adjust the shape in order to fit perfectly like a glove in a hand in that active site. And finally, they produce a product. The enzyme substrate complex produce a totally new molecule. Sometimes only one, sometimes more than one molecule produced, and this one is called, considered a product. When after all the process, the enzyme can be reused on and on and on again because the enzyme never lose the original 3D shape in the process. At the bottom of this slide, you can visualize the larger portion, that is the blue color, represent the enzyme, and the enzyme has that specific active site. And this active site is produced or is made for specific substrates. In this case, the substrate A is green, the substrate B is pink. They use that active site. When they are using that active site, there is a molecular bond between the substrate with the enzyme. But after all, the final product, that could be a product AB or a product C and D, or different kind of product. Could be only one, could be more than one, but certainly the product is totally different molecule that the substrates are coming from. The enzymes are also named in one particular way, that they have, all of them, they have a suffix that finish in "ase", like you read in this slide: phosphatases, synthetases, hydrolases, dehydrogenases, kinase, isomerase, but all of them has the same suffix, ASES. As we mentioned before, the names is also another way to determine the function of the enzymes, and sometimes the same enzyme is found in more than one organ and they have similar functions, and these are now called isoenzymes. It's the same enzyme but is located in different organs. Also, that name of the enzyme is the one that determines the function, and also the measurement of those enzymes are used to detect and diagnose certain diseases. For example, the acid phosphatase is used to diagnose certain levels of prostate cancer, or the amylase. Amylase is produced mainly by the pancreas and is measured to diagnose pancreatitis or pancreatic cancer, or sometimes a perforated peptic ulcer. Uh, Aldolase. Aldolase is another enzyme helping in diagnosis the muscular dystrophy, or the lactate, the hydrogenase that is used to measure or to diagnose a myocardial infarction, liver disease, renal disease, pernicious anemia, and similar diseases. So the levels of certain enzymes are helping to diagnose certain diseases. The rate of activity of one enzyme is measured by the rate at which substrate is converted to product. So, in other words, the enzyme can be exposed to high activity, normal activity, lower activity, and everything in the middle. This enzyme activity is highly influenced by certain effects. One is temperature, the second one is the pH where the enzyme is founded. Also, the enzyme activity is affected by the concentration of cofactors and coenzymes. It's also affected by the concentration of this enzyme and the concentration of the substrate used by this enzyme, and possibly stimulatory or inhibitory effects of products on enzyme function. Means that how much we need that enzyme or how much we don't need that enzyme. So all these factors are the ones that influence the activity of that enzyme. As I mentioned before, temperature is a very important factor that affects the activity of the enzyme. Considering that we are talking about the human being body, we have an internal body temperature that have to be constant, and our body has different homeostasis mechanism in order to keep that internal environment constant at 37 Celsius or 98.6 Fahrenheit. Certainly, that temperature we mentioned before, that is affect the enzymes activity. Why? Because a decrease in internal body temperature decreases tremendously the production or the activity of this enzyme. On the contrary, if our body temperature increase above the normal level, increase the effect of this enzyme activity. However, that increase in temperature after passing certain levels that we review this concept in homeostasis is totally opposite action, is inhibit the action of this enzyme. For example, if we are exposed to fever, if we are exposed to 38 Celsius or more, that enzyme activity decreases the function. But on the top of everything, if we develop a very high temperature that is 39 or even 40 Celsius, eventually that enzyme, like any other protein, can be exposed to denature. In the nature, that protein, an enzyme is a protein, change the 3D shape. Changing the 3D shape, that enzyme cannot use the substrates and cannot produce the product that is expected to find. pH of the environment where the enzyme is located has a very important impact in the enzyme activity. For example, there are enzymes located in our stomach, another enzymes located in our mouth, another enzymes located in our intestine, other enzymes located in the duodenum, other enzymes located in the vagina, and the list is really very large. But certainly, the optimal activity of the enzymes located in the stomach have to work ideally in that pH. If we take this enzyme from the stomach and transport it, if just metaphoric conversation, and we can transport it within the mouth, that enzyme cannot produce the same action because the pH in the mouth is a pH 5, around 5, and the pH in the stomach is pH 2. Both are acidic, but are different pH. So that enzyme that is working in the stomach is working beautifully under the pH 2. And you have certainly three types of enzyme. The pepsin that is produced in the stomach. Pay attention. Pepsin is a product that is coming from an enzyme called pepsinogen, but is produced within the stomach under pH 2. We have the salivary amylase. The salivary amylase is around pH 5. In this graphic, you see the amylase, salivary amylase is activity is produced under pH between 6 and 7. And I totally disagree with that. And I can tell you with high knowledge, because I'm a dentist, that the salivary amylase is working in a mouth pH that is average pH 5. It's always acidic, but it's never alkaline, certainly. The other type of enzyme is the trypsin, and the trypsin is located in the duodenum. Certainly, I agree with that. Yes, duodenum pH is alkaline, is highly alkaline, between 8 to 9 pH. And this one is a graphic how these different environment and different pH are ideally for these three types of enzymes. We have also coenzymes, and coenzymes are organic molecules derived from water-soluble vitamins. You will expose to different coenzymes in our physiology, and the one that you will hear really very soon is during the glucose oxidation or cellular respiration that probably you remember from a previous subject like is organic chemistry, how we oxidize the glucose, and in one of the process is called the Krebs cycle or citric acid, where we use the acetyl coenzyme A. A coenzyme is derived from a water-soluble vitamins, and the function of them is transport hydrogen atoms um, in that environment. Cofactors are another molecules very important, and they have also a very important role. Not all the enzymes are working with cofactors, but most of the enzymes are working with cofactors. A cofactor is a molecule, in general are metal ions such as calcium, magnesium, copper or zinc, but the function of them is to help that bonding between the substrates with the enzyme in the active site. This one is the function of the cofactors. So when the bonding between the substrates or reactants with the enzyme is stronger, facilitated by this cofactor, the function of this enzyme is even better and the product is even more precise. You also have to know that several enzymes in our body are active already. They are ready to be used, but another one are inactive, and when an enzyme is inactive, it's called zymogen. It's an inactive enzyme that need to be activated in order to produce the function. One simple example is the pepsinogen. The pepsinogen that is produced by the chief cells in the stomach under the pH2 produced by another cells, the parietal cells in the stomach that produce hydrochloric acid. That pepsinogen that is an inactive form by the production of this acidity pH can be activated and converted in pepsin. That pepsin has the function to digest a very small portion of proteins in our stomach. From the proteins we ingest in the food, only 20-25% of those can be digested and absorbed at the stomach level. Just 20-25%. The other roughly 80% is digested in the following portion of the digestive system, that is the jejunum. The small intestine, passing through the duodenum, getting into the jejunum. Now, another important concept is that the enzyme activity can be inhibited. If we have enough amount of product that is desirable for our physiology, there is no need to continue producing the same product. So that enzyme have to be inhibited. Substrate concentration is another factor that increase or decrease the enzyme activity. Obviously, if we have less supply of substrates or reactants, decrease the activity of the enzyme. We don't have the ingredients to produce that product, we cannot produce the product. But on the contrary, if the substrate concentration increase, eventually increase the enzyme activity. However, if this substrate concentration reach the maximum rate that the enzyme is capable to use, that enzyme becomes saturated. And when the enzyme becomes saturated, the amount of product, it doesn't change, remain in that high rate, but not above that normal rate. In other words, there are substrates in the field that is not used. What is reversible? What is a reversible reaction? A reversible reaction is the one that can proceed in one direction and can be reversible to the opposite direction where it was coming from. We talk about the carbonic anhydrase, that is one important enzyme that is located in our erythrocyte. And I invite you to watch the video I'm going to include in this playlist to understand that the carbonic anhydrase use different substrate depending with the capillary we are talking. So we have basically, regardless the type of capillaries, we have basically two main capillaries, the ones that are located all our body called systemic capillaries, and the other capillaries that are located around the alveoli. Now, this carbonic anhydrase is mainly located in large amount inside the erythrocyte, and inside this erythrocyte is using different substrate if we are talking about the systemic capillaries or we are talking about the alveoli capillaries. And I invite you to watch the video to clarify even more about these substrates. In previous chapters, we talked about the same topic, and I invite you to watch the video to strengthen that knowledge. Now, in a metabolic pathway or a chemical reaction pathway, because metabolic is a chemical reaction, and yes, we have billions of or trillions of metabolic pathways in our body. Now, if we talk specifically about the enzymes, that metabolic pathway can be in two types. Could be linear or could be branched. The linear are not the most common, but certainly it happens when you have an initial substrate that use one enzyme, only one substrate use one enzyme, sometimes use more than one enzyme, like is, if you remember from anatomy, in the duodenum we have three types of enzymes, the trypsin, chymotrypsin, and carboxypeptidase or carboxypeptidase. Now, these three enzymes are the ones are used by the substrates that are basically monosaccharides or disaccharides in order to produce a final product. And this one is a linear pathway. Again, we use only one substrate. Probably we use one enzyme or more than one enzyme to produce a final product. The other type of metabolic pathways using enzyme is a branched metabolic pathway. In the one, yes, we use the initial substrate. That could be only one or could be more than one, but certainly the pathway is branched in two different ways to produce the final product. And this one is the most common metabolic pathway that is used in our body. Finally, if we talk about the enzyme activity, also is affected by the final product. If the final product is enough for the need of the body at that time, at that moment, obviously we have to slow down the production of this enzyme, and how we produce that reaction is inhibiting that enzyme to release less product. In the process called allosteric inhibition, the product binds to the enzyme at a location away from the active site and change the 3D. In other words, the product changed the 3D conformation of the enzyme. And if the enzyme changed the conformation, cannot produce the same amount of product. Now, what is the function of that? Is reducing or avoiding the accumulation of product that is not needed at that time, at that moment. In this graphic, you have another sample of the product inhibition. In other words, if we have enough amount of product, after all, that the body or the cell doesn't need anymore, automatically produce the 3D shape change in the enzyme in order to don't allow the production of more product that is accumulated. It's avoiding the accumulation of the product. If the enzyme is a protein, the protein code is encrypted inside the DNA. And if there is an alteration of the DNA, either because it's genetically passed from generation to generation or is an abnormal cell division that produces a change in the DNA composition, will have eventually a production of enzymes that are not normal. And that enzyme that doesn't have the proper 3D shape, the proper chemical structure, result in different, it's called errors of metabolism. And these errors of metabolism can be result in different disorders like mental retardation, epilepsy, skin cancer, diarrhea, hypoglycemia, muscle fatigue and pain, different kind of disorders that we develop after birth. I mentioned at the beginning of this PowerPoint that the enzymes are related to energy. Why? Because it's a special molecule our body uses in order to trigger a chemical reaction faster but with less amount of energy. Now, because we talk about energy and how energy is related to our body, it's time to define the two laws of thermodynamics. The first law of thermodynamics is pretty simple. Means that in our planet, and obviously we are part of our planet, the energy is never created and never destroyed. It's passing from one point to another one. It's passing around. So in some places the energy is produced, but that energy is used by other chemical reactions. Basically, the first law of thermodynamics mentioned that the energy is not created or is destroyed, it's transformed. In that transformation is not 100% in energy. There is waste material as well. The second law of thermodynamics is expressed in one particular topic that is called entropy. Entropy is defined as a disorder of a closed system, and that this disorder is always increasing as the chemical reaction proceeds. Now, if entropy is defined as a disorder, more entropy is generated. Meanwhile, more entropy is generated, less energy is available to do work. Pure energy is available, but is not available to do a work. Now, humans are not a closed environment, and we are exchanging heat with the surrounding or our external environment, and we decrease the entropy at the expense of the entropy of the external environment. So, spontaneous processes generate entropy. This law relates to endergonic and exergonic reactions because basically there are two types of reactions in our body. One are called exergonics and the other one are called endergonics. And I invite you to watch the additional videos. Specifically, this one is talking about endergonic and exergonic reactions. Broadly, we used to say that the endergonic reactions, in order to proceed, need to absorb energy from the surrounding in order to produce that chemical reactions. The reactant, and that one is a common denominator for all the endergonic reactions. The reactants of substrates contain less energy than the final product and occur in reactions where the reactants are at a greater state of entropy, means that the disorder of the reactants is higher than the product. For example, the plants that need to use energy from the light to turn carbon dioxide and water into glucose. This carbon dioxide and water, they have higher entropy or higher disorder. The final product is glucose, and glucose has less entropy or less disorder. On the contrary, exergonic reactions are chemical reactions that in the way that they proceed or in the pathway of this chemical reaction release energy. And this one is the beauty between endergonic and exergonics. That exergonics release energy, they don't produce energy, they release energy, and that energy release is used by the endergonic reactions. In these exergonics are totally opposite than the endergonics. The substrates or reactants is a bigger molecule, contain more energy than the product after that chemical pathway. And the product will have less free energy than the reactants itself. For example, the production or the breaking of glucose. When glucose is breakdown to produce energy that is in the way of ATP, in all that chemical pathway where glucose is breakdown is releasing energy. However, glucose is a molecule with very low entropy. And the product at the end, that is carbon dioxide and water, has way higher entropy. In the way to release that energy is called exergonic reaction and is increasing the entropy. Another sample that we can mention about exergonic reactions is when we have a molecule of ATP that is breakdown into other two molecules, ADP and inorganic phosphate, and I will explain pretty soon. In this graphic, you have the sample I mentioned before. The substrates for the glucose oxidation is one molecule of glucose that you have at the top of the ladder and 6 molecules of oxygen. These substrates has a higher energy than the final product, after all, at the bottom of the ladder, that are 6 molecules of carbon dioxide and 6 molecules of water. Also, the substrates at the beginning has a very low entropy, and the products at the bottom of the ladder has a very high entropy. And this one is a sample of exergonic reaction. During this semester, you will hear about different chemical reactions, and I invite you to be solid in your knowledge right now in order to get familiar later on in the semester, because otherwise the following concepts are going to be more complicated, and if you have to catch up, probably you don't have the chance to be above the water. But that chemical couple reactions are using energy from the environment and are broken down in exergonic reactions that drive another endergonic reaction. It's important that keep these two elements in balance between exergonic and endergonic. The exergonic reactions are the ones that release energy, and that energy release is used by the endergonic reactions to proceed. For example, I mentioned before about the ATP. The production of ATP is an endergonic reaction. Why? At the bottom, in letter D, you see that when we add ADP plus inorganic phosphate, we produce ATP. Now, these two substrates has less energy, higher entropy, and the final product, ADP, has higher energy and is a sample of endergonic reaction, because in order to add ADP plus inorganic phosphate, we need to absorb energy, and this absorbed energy is kept in that final product. That's why the final product in the endergonic reaction has higher potential energy. On the contrary, in letter E at the bottom of the slide, you have a sample of ATP. The ATP in an exergonic reaction release energy. When we hydrolyze the ATP, we produce one molecule of ADP plus inorganic phosphate. Again, a sample of exergonic reaction is when the substrate, in this case ATP, has less entropy and higher potential energy. And in the process to produce the chemical reaction, that is ADP plus inorganic phosphate, release energy. In other words, these two products, ADP plus inorganic phosphate, they have higher entropy than the original substrate, that is ATP. And this one is example of exergonic reaction. Like any other biological topic or subject, this one is not exemption. The couple reactions need to be organized in order to know which one are the types that we can have of those. As I mentioned before, one way to organize the chemical reactions is by if they absorb energy or they release energy. And because of that, they are organized in exergonic or endergonic. Another way to organize the chemical reactions is by couple reactions, the ones that are called, they are calling different names, but one typical couple reaction is hydrolysis and condensation. Condensation is also called dehydration. So hydrolysis and condensation or dehydration is one type of chemical reaction. By hydrolysis, one molecule of water breaks the bond between two molecules and produce two smaller molecules by hydrolysis. By condensation is the opposite. We use the little substrates that we bond together, and the way that we bond together is releasing one molecule of water. This one is dehydration. We mentioned when we talk about the organic molecules, any type of those, they are built by monomers. So if we add monomer by monomer, we use dehydration synthesized. That is the way how to build up a macromolecule. On any of the kind, could be a carbohydrate, a protein, a lipid molecule, or a nucleic acid. On the contrary, by hydrolysis is when we break down a macromolecule into the monomers that are composed. Another type of couple reaction is also called phosphorylation and dephosphorylation. By phosphorylation, we add an inorganic phosphate, and by dephosphorylation, we split one inorganic phosphate. Example of phosphorylation, for example, is I mentioned before, if you have one ADP molecule and you add one inorganic phosphate, bonding together, they produce a bigger molecule that is ATP plus water. And this one is also a sample of endergonic reaction. By dephosphorylation, it's on the opposite way. You have one ATP that you add one water molecule that split the bond or break the bond, and we produce ADP plus inorganic phosphate. So pay attention that the case when we split ATP into ADP and inorganic phosphate is a sample of hydrolysis reaction, because it's what really happened. Another couple reaction is called oxidation reduction. The previous two couples, one is hydrolysis dehydration, the other one is phosphorylation dephosphorylation, doesn't happen simultaneously. It can be reversed back and forth, but never happens simultaneously. In that chemical reaction, oxidation reduction, that chemical reaction happens simultaneously at the same time. And it's one sample or one type of a couple reaction, but also is an exchange, because in that molecule, one atom is receiving one electron, and this is considered reduction. Is one atom is absorbing the electron, is reduced. And the atom that is losing the electron is called oxidized, and happens simultaneously. This typical reaction, that is oxidation reduction, is commonly found in the last step of glucose oxidation, that is called electron transport, when the atom is passing back and forth from one atom to other atom, and the last one who is reduced is the oxygen that is reduced, is receiving the last electron in that electron transport. In this slide, you have another sample of oxidation reduction, where obviously the atom that is losing the electron, the one that loses the electron, is called oxidation. The one that receives the electron is reduced. We use the same terminology, oxidation and reduction, when the passage, instead to be an electron, is hydrogen. If that molecule loses hydrogen, that molecule is oxidized, and the molecule that gains an hydrogen is reduced. Typical molecules in our body that move hydrogen like carrier molecules is the NAD and the FAD. The NAD and FAD are another type as nucleic acid. Nicotinamide adenine dinucleotide is the NAD, and the FAD is the other very important nucleic acid composed by these particular nucleotides. And both of them are coming from different type of vitamin B. And these molecules are the ones that are very common to be found in the electron transport or during the Krebs cycle. Like they are mainly products after the Krebs cycle, and these NAD and FAD are the ones that are going to exchange hydrogen. In other words, can be oxidized or reduced. And basically what the function is to transport energy within the cells that finally produce the ATP. So, reinforcing the previous slide, NAD in this case, and the same happened with FAD, is they can absorb a hydrogen and become reduced, or the same NADH that is reduced can lose that hydrogen is become oxidized. And with this topic, I invite you to watch the following videos that support the material in this PowerPoint with a little more explanation, and hopefully it's going to be much easier for you to break down this material.